So the sad reality, oh, not the right slide. This is not the right slide. Oh, there. So the sad reality is that almost half of cancer patients fail to respond to their first treatment, resulting in the tragic loss of nearly 10 million lives every year. And this isn't just tragic, it's avoidable. I'm Ana Teresa, CEO and co-founder of Express Tech. We're on a mission to save lives by transforming treatment decisions. The problem is simple but crucial. Most advanced therapies target specific mutations found on tumor proteins, whilst all treatment decision relies on DNA testing. So what's missing? RNA, the molecule that bridges DNA onto protein. And the game changer is that we have shown that many DNA-positive tumors actually fail to produce RNA, meaning that all of our competitors are currently sending many patients to targeted therapies from which they do not benefit but still feel the side effects. So what's the solution? To test RNA. At Express Tech, we're developing the first RNA technology empowered by AI to accurately select patients for targeted therapies. And our first product, Express Peak, tests for the most common mutations found on breast tumors and sets the scene for our larger vision of a platform testing multiple mutations across all cancer types. Our innovation is simple, fast, and precise, constituted of kits of reagents sold to clinical labs and hospitals, but most importantly, a cloud-hosted AI software that generates a medical report that empowers oncologists to take the best data-driven treatment decisions. The market opportunity is immense with our first application as a treatment selection tool, but our target market for our first product is actually quite conservative. If you consider all of the multiple other applications that our products have, including clinical follow-up by testing liquid biopsies. We are set on a strong IP strategy and plan to launch as research use only tool next year, so we're also looking here for partnerships for whoever wants our help to improve the design of their clinical trials. And we are a female founding company of successful scientists, and we recently enlarged our team with marketing and business skills. We're raising our first private round of investment to supplement public funds we already have and launch not only just as RUO next year, but initiate the certification process for launching as diagnostics in 3 years. But this is so much more than just about diagnostics. This is about the future of cancer care. So join us in rewriting the rules of clinical testing to make personalized cancer care a reality.
Hello, Karina from Fund F.
Oh, hi.
So my first question would be, how does then the user journey look like? Could you maybe give an example on how the oncologist will change the treatment? Like, what's an example on how I could adjust it?
The thing is that, like, the drugs that already exist and are in the market, they're good. The question is that the oncologist, once a patient is diagnosed, then they have to use these tests, companion diagnostics, to actually decide the line of treatment based on guidelines that are established by associations like ESMO and ASCO. But the thing here is that they have many different drugs to choose from, and they need good and precise tests like ours to actually put their patient on the most correct therapy right from the beginning, because every month that a patient is on the wrong or unresponsive treatment, it's 10% less of survival chances. So it's important that we get the patients right at the beginning, and we are improving the success rate of the response to treatment.
Hi there, really enjoyed your talk. Peter Liu, medical doctor and molecular biologist, CEO of Oxford Cancer Analytics. So obviously you talked about the disconnect between DNA, RNA, and protein. Mm-hmm. Why not just look at DNA and protein directly to look at expression? And what are your thoughts on some of the disconcordance between RNA and protein expression, especially given some of the negative feedback loop that exists?
So the question here is that, like, this is based on almost 2 decades of research. And yes, actually, the target of the therapies is proteins. Proteins are not as easy to work with as is RNA. Maybe they suffer from a little bit of bad reputation, as RNA did, until it actually got 2 Nobel Prizes in the last 2 years, and they were the first ones. So using nucleic acid is cheaper, is faster, and there's one thing about RNA and about our biomarkers that is very important, is that we are actually fully quantitative. And with proteins as well, it's more difficult to actually quantify things, and our biomarkers are actually like quantitative in the sense of, for example, blood pressure, in terms of risk for cardiovascular disease, for example. So based on the levels of these RNAs with mutations, we can actually also provide prognostics.
Anna Teresa Maya, thank you very much.
Thank you.
Thank you. Here we go. Next up, Francesca Domenic-Wódka, PhD, founder and CEO at NEN.
Thanks for the opportunity to present today. I'm Francesca Wódka. 400 million kids are impacted by pain. These kids have very few options to help them manage that pain. Our focus is on pain in pediatric cancer, where nearly all of these children experience pain. They also are at very high risk of medical trauma, and these negative pain memories can be quite sticky and can persist through survivorship and into adulthood. In fact, 59% of adult survivors of pediatric cancer have pain in adulthood. Children communicate about their pain very differently than adults, so it's hard to know how to treat these children. And there are too few therapists to treat these kids. Most families are told they have to wait between 12 and 18 months to see a pain specialist. Despite this huge unmet need, very few solutions exist for kids. That's what we're endeavoring to do. Kids matter, and we're committed to kids. We've developed digital solutions for kids by gamifying cognitive behavioral therapy. We know CBT works, we're just digitizing it at scale. We've created 3 virtual companions. Dolores delivers CBT related to pain, and then Sarah and Tony are more focused on the serotonergic receptor pathways on mental health. But it's not just the kids. We've spoken to a lot of parents throughout this journey, and the one thing that's been overwhelmingly consistent is their sense of helplessness. There's not a lot that they can do when their child is in pain. So we've created a suite of services specifically for parents to help them help their kids get the best outcomes out of the therapy. And this is how we're going to get it to them. We're really excited to have recently launched in the US, UK, Spain, and Portugal, in English, Spanish, and Portuguese. But key to our project is trust, and that trust comes from evidence. We're generating evidence across some of the top centers across US and Europe, like Stanford, who will be running our clinical validation study for us. These are the hospitals that will ultimately share the word about NEN with the families, and also eventually, once we're a regulatory cleared and prescribed product, will be the ones prescribing NEN. In addition to our pain platform, we're also developing a standalone mental health platform as well. Very much aligned with our mission and vision, we're looking to democratize pain management at scale. We currently have a buy one, share one program, the kind of Tom's shoes of digital health, where for every license that's purchased by a family, we allocate one to a family who may not be able to afford it. We are really excited to have been a part of several accelerators, as well as have won some awards over the 2 years that we've been founded. In addition to our amazing clinical team that translates what works in the clinic to a really fun and engaging platform for kids, we have these great advisors. Our clinical advisory board are experts in treating pain, and our business advisors are pediatric innovators, social impact drivers.
Sorry, we're going to have to stop you right there. Apologies. It's time to move on to the questions. 2 minutes for questions.
Thank you, Francesca. Can you tell us a little bit of the outcomes that you expect with this product, and whether they go beyond the pain and could be implied somehow on the progression of the cancer for kids in that case?
Yeah, it's a good question. So we're using the same metrics and outcome measures that are used for CBT studies, and they're all already embedded in the platform. So every child that joins the platform essentially becomes a clinical subject, and we can track their results. So we're checking their pain, how they're managing their pain over time. There's also something importantly called pain catastrophization. So it's the parents' influence on how their reactions to the kids' pain are interpreted. On our parents app, we have a pain catastrophization scale for the parents as well as for the kids. And we're also looking at measures of anxiety and depression. With regard to outcomes, there are a number of reasons that drive profitability for hospitals. You can't discharge a child if they have unmanaged pain. If a child is really anxious about a procedure because of the pain that they have, it requires a lot more staff. So part of the clinical validation study will include some of those pharmacoeconomic metrics as well.
Thank you.
Very quick question, only 10 seconds. So.
Thank you very much. How about the VC, the investors that you already have or you are looking for?
So we are currently raising a $2 million seed round. We have 1.1 million already invested from family offices, venture capitalists, some of our advisors and experts have invested money as well.
Thank you very much.
Thank you very much, Francesca. Andreas Hallner, President and COO of Oxford Cancer Analytics.
The problem is that the deadliest cancers are diagnosed too late because we rely currently on symptoms and invasive approaches. Over 50% of all cancers are detected at late stage, at which point current treatments can't cure them and can be up to 3 times more expensive to treat. OxCan solution is a simple blood test able to detect materials predictive of cancer in a safe, inexpensive, and routine way. Unlike many competitors who conventionally rely on DNA-based approaches, which are limited for early detection sensitivity, our unique proteomics approach enables us to double sensitivity for 8 types of cancer, and we can do this at 1/10 the cost of manufacturing and in a scalable way compared to competitors. We're beginning with lung cancer, the biggest cause of cancer-associated death worldwide. The market is large, unfortunately, 217 billion euros, and we're starting with the largest subsegment, screening. Our performance is transformative. On a cohort of over 600 patients, OxCan detected over 90% sensitivity and specificity for early-stage lung cancer, compared to just 20 to 48% for competitor companies. We've built international biobanks spanning 3 continents and are working with leading academic institutions and healthcare systems, including the NHS. In 2025, we're launching the first lung cancer detection product as a laboratory-developed test with support of leaders internationally in global diagnostics networks, including direct investment from Burfin, LOIs signed from Randex and Alpha Laboratories. And then we're scaling to full regulatory approval in 2026 with independent test kits for lung cancer, ISO 13485 already obtained, and we're currently in the process of FDA breakthrough submission, and scaling in 2028 to multi-cancer detection. By 2028, we want to help over 120,000 patients per year, with annual revenue exponentially growing and at over 50 million euros. So what's our secret? Well, we have a unique proteomics approach looking at thousands of proteins per patient and a specialized machine learning framework which can distill that into a succinct panel of 10 to 30 top proteins, which will translate into an inexpensive immunoassay test kit, which can be used across the world. We've got unprecedented performance compared to competitors, 90% sensitivity and specificity because of our proteomics approach, our early cancer focus, and our affordability. And our team has diverse expertise. Co-founder and CEO, Dr. Peter Liu, physician scientist with over 10 years cancer research experience. His lab won the Nobel Prize. Myself, Heinrich, our SVP, has already brought multiple lung cancer products to market. We've just finished a Series A of $11 million, and we have a second window open now for value-added investors. We'd love you to join our journey to impact the lives of millions of patients and their families worldwide, and we'll be in the booth at S55 outside today to speak. Thank you very much.
Two minutes for questions.
When you say low price point, so how much do you sell this for?
How much do we sell this for? So the price point depends on where we are. It's going to be between 100 and 250 pounds sterling, depending on the particular part that we're in the world. And we're making sure that it's very affordable in, say, the developing world as well as also for America, because we've got different reimbursement models.
Thank you for, shall I go? Thank you for the presentation. I was wondering at which regulatory stage are you and how close to certification, if so?
Yeah, so for FDA approval, we're currently in the process of submitting breakthrough designation. So that's that, and that's for full approval for lung cancer, and then we're doing that in parallel for multi-cancer as well thereafter.
And by when do you expect the approval so that you can
2026.
start? Okay, good. Thanks.
Any other questions?
Andreas, good talk. Just to emphasize, why lung cancer again?
Yeah, I mean, lung cancer is the biggest cause of cancer-associated death in the entire world right now. It's where you have the most need for patients. There is no screening test for lung cancer right now. That's what we want to change. We want it to be the case that we can cure lung cancer through early detection on a massive scale.
Quick question.
Have you thought of doing it in other fluids besides blood?
Yes, we have. But that's also part of our R&D. But we want to make sure that it's scalable and it can be deployed without having further infrastructural needs and training needs to make it really accessible to patients.
Andreas Hallner, thank you very much.
Thank you.
Next up, Marco de Boer, CEO, Predica Diagnostics.
Hello, my name is Marco, Predica Diagnostics. We are developing precision diagnosis for women's health. Our first product is aimed at cervical cancer prevention. And cervical cancer is caused by the human papillomavirus. And every year, more than 600,000 women worldwide are diagnosed with cervical cancer, and only 50% survive. And this is completely preventable with appropriate screening and vaccination. For example, 2 weeks ago, a Dutch singer was diagnosed with cervical cancer for only 28 years old. Current screening programs consist of 2 steps. So the first step is a PCR to detect the HPV virus, and all HPV positive cases are then screened with cytology. And the problem is that the cytology is not specific enough. And this results in lots of late diagnosis. For example, there are still more than 2,000 deaths in Holland every year, more than 500,000 false positive cases. And if you do the calculation, there's more than 500 million euros in unnecessary healthcare costs. So with Predica, we are developing a test that will replace the cytology. We call it the CervicIDX test. And we will reduce the amount of women that need to go to the gynecologist. We'll be able to have a better prediction of cervical cancer and save lives and a lot of costs, of course. So a bit how our technology works is that we have developed what we call circular DNA probes. And with these circular DNA probes, we can catch the RNA molecules that we find in cervical scrapes. And in this case, from the HPV and from biomarkers. Our readout is based on sequencing. So we generate all the sequences of the HPV virus and the biomarkers. And we have developed our own proprietary algorithms to be able to predict the cervical cancer risk. In general, our product is based on an assay with which you can detect the HPV and the algorithms that we can use to predict the risk. Our technology is scalable, so we have developed our product for cervical cancer, but we are also looking at the microbiome and endometriosis, for example. And I think I need to close it down. So what we are doing now is that we are developing our products for research use only. We are marketing that at the moment. And we are now opening an investment round of 4 million euros, of which 50% has been committed by our current investors. And that will allow us to develop our product through IVDR and CE and be able then to offer this to patients. So if you have any questions, please let me know.
Sorry, maybe I missed it. So what kind of biomarker do you need? So what sample do you need to draw from the patient?
So we use the general scrapes that are currently taken during an exam. so in the same buffers. So our assay fits into the current testing procedures.
What's your business model?
At the moment, we are selling our kits to research institutes that work on cervical cancer, and we are collaborating with IVD players to co-develop and co-market our technology.
Thank you, Marco, for the presentation. Can you tell us a bit about the power of your test compared to current diagnostics?
Yeah, of course. Yeah, that's a good one. So, like the previous speakers mentioned, current tests are all based on DNA. And for example, with cervical cancer, you know which HPV is present. But we are looking at RNA, so we also know which HPV is really active, and we also measure which biomarkers are active. So we have a much better specificity and sensitivity to predict cervical cancer.
Okay. Do you have by chance the specific numbers of the specificity and sensitivity compared to current diagnostics?
Yeah, so the current cytology is only 50 to 60% specific. So that's why a lot of countries are skipping cytology. Our current assay is around 88, 90% specific. And of course, we want to get it higher, but we are much better than the current standard.
Thank you.
How easy is it to integrate this within the current diagnostic pathway, and what feedback are you getting from clinicians on that front?
Yeah, well, our test runs on the current scrapes, so therefore the patient and the gynecologist, there's nothing different. The laboratories that are using our test can do this on current equipment. The only thing is that they need to use sequencing equipment.
Okay,
And that's
Marco, we hate to stop you right there.
No problem.
Apologies, not personal.
Yeah.
The time is up for the questions. If you would like to ask further questions or get a completed answer, you can do so in the trade show outside. So thank you very much.
Thank you.
Round of applause.
Thank you.
Thank you. Thank you. And the final presenter for this part of the track, Omar El Nahas, CEO of Stratify. Omar, and then we'll have a chance, everybody, for an extended break before the next track. So if you don't get your questions in, you'll have time.
Perfect. Thank you so much. Hello, everyone. My name is Omar. I'm CEO, co-founder of Stratify. And at Stratify, we develop the next generation of cancer biomarkers for precision oncology. And our founding team consists of computer scientists, an oncologist, and a radiologist. And in the past years, we have defined the pathway of defining and discovering novel biomarkers using AI. And if you have heard about computational pathology in the recent years, it's likely because of us. Because in 2019, we published our landmark study in Nature Medicine, showing that we can find a correlation between phenotype and genotype from routine medical imaging. And since then, we have published more than 100 articles in venues such as Nature Medicine, Nature Cancer, Cancer Cell, and even in the last month, we published in Nature Protocols and in MICAI technical conference. And the thing that we are focusing on, the problem that we're tackling, is that treating a cancer patient is super difficult. Also what we've heard from the previous speakers before, because there are so many drugs on the market and it's continuously evolving. New drug, new drug, new drug. Which one are you going to give to your patient? And this question can only be answered by having better biomarkers. And this is exactly what Stratify develops. We develop novel cancer biomarkers, stratifying patients between responders and non-responders. And we target two specific applications. One for existing drugs that are currently already on the market, where we are being reimbursed by insurance and distributed through hospitals, where we've already signed several pilots, are doing trials, clinical studies, and collecting thousands of patients and their data. At the same time, we are assisting in drug discovery, where we're working together with pharma and biotech in order to bring new drugs to the market, both in the preclinical and clinical setting, where we've already have close to 2 million in projected ARR since our pre-seed round only 3 months ago. Our core technology builds on the multimodal representation of a patient, data that is usually lost due to their complexity. We can correlate patient survival with the multimodal properties of the tumor, both in the spatial, but also on the DNA and RNA level. with our core technology, we can therefore develop multiple applications. For example, for drug response, for drug development. And one of our highlighted applications is the risk of recurrence in breast cancer. In our study, which we validated on more than 6,000 patients together with Memorial Sloan Kettering in the US, we show that we are beating the clinical standard, the standard clinical assessment by 48%. And if we're looking at the status quo test, Oncotype DX, which is currently on the market, we are beating them in predicting the actual recurrence of a distant metastasis in breast cancer by a huge margin. All the while, we can do it instantly, instead of waiting 5 weeks on the results. And this is just the beginning. We are funded by a very strong team of investors, and we are expanding not only in breast cancer, where this specific test was a 300 million annual market.
Okay.
We're expanding.
Omar, very, very, very sorry.
Sorry, all
So
good,
sorry.
all good. Here you
Accept
go.
my apologies.
No,
So we've got 2 minutes now for questions.
Perfect.
Okay?
Yeah.
Hi, Omar. Peter and Leo again. I'm asking too many questions. Great talk. With your validation result, 6,000, was it a holdout? Was it a prospective study? Was it blinded, or was it discovery training set? Just more information on the validation.
This was all retrospective because we are looking at a follow-up of more than 12 years, because for breast cancer, anything before 5 years is basically useless information because the recurrence can start later. So this is all retrospective. At the same time, we've already started prospective but observational studies at the moment to use our tool in parallel.
And the result you show was a holdout validation with an independent blinded validation, and also what was your primary and secondary outcome?
So this was independent holdout validation. So this was unseen, blinded. And what was the second question?
Was with primary and secondary outcome, or was it blinded? Was it 20% holdout of the 6,000 cohort?
Progression-free survival was the primary endpoint.
Okay, of all common histologies within breast cancer, what is specific?
So this was specifically in hormone receptor positive, HER2 negative early stage breast cancer.
Okay, got it.
For
And
the
also,
adjuvant setting of chemo.
What % of holdout was it out of the 6,000 cohort?
So this 6,000 was the holdout set.
Okay, and you had a separate discovery set.
Yes.
How big was that?
5,000.
Okay.